The combination of HES and 3D organoids creates a powerful platform which allows to study many conditions and many images taken from each condition. In this second use case, we want to show you how Amira handles multi-channel HES images of organoid models by extracting 3D information such as segmentation and penetration of immune cells into the spheroids. Rosa, let's dive into Amira and your demo.
Thanks. And let's go for the presentation of the second demo. So here, again, I want to show you the entire project. On the left side, this is, again, the complete workflow. So you can see different modules again. And then on the right side, you can see different visualization. So basically, on the top, we have a graph which represents the number of T cells. It was calculated the population of the T cells. And then on the right side, this is a distance map, which basically it was created to represent the distance inside the spheroid. And then on the bottom side, we have other two visualization. So on the left, this is the volume rendering for the 3 channels. And then on the right, this is the population of the T cells into the spheroid. So basically, we want to represent the penetration of T cells into the spheroid. So let's start in Amira software from the beginning. So I'm going to open a project. And first of all, I want to focus on one module, which is the HS Plate Manager module. Okay, so here we have our project, and this is the HS Plate Manager. So the HS Plate Manager is a very useful module because it enables loading, visualizing, and processing multi-channel fields. And the data must be provided as a folder containing DIB files. So here you can see in the plate directory, we should specify, indicate the directory with a folder containing the DIB files. Once that the files are uploaded here, then you can get here the plate information. So in this case, it's 2 wells, 2 fields, 3 channels. And you can also decide in which mode you want to load this sample. So load field, this means 1 field and multi-channels and multi-channel data. Or you can also, for example, process the entire plate or stitch fields from well. And then you can also decide wells and fields. So if I press on apply here, so now I can see the data, which correspond to 1 field and 3 channels. I can link, for example, an orthoslice or, for example, a volume rendering. And then if I go here, I can also adjust the intensity for the 3 channels. And then, yes, you can get, so basically the red channel correspond to the cancerous cells. The green channel is the T cells, and then the staining of the nuclei is in blue. You can also go in the volume rendering here, and you can deselect, for example, all channels, and then you can select only the one that you are more interested, for example. It is also interesting that we can here run a recipe. So if I click right and choose for create object, I can type recipe player here. And then I need to decide, so upload the recipe that it was saved. So I go here and then I can go for spheroid. Basically this recipe, so sorry, here we need also to select the data input and then you can press on apply. So this recipe was created by the expert in order to study the population, the samples of the cancerous cells. So if I link here an orthoslice, you can see the threshold of the cancerous cells. And then the label analysis, this is the population of the basically of the cancerous cells, so volume 3D and the area 3D. Okay. So now if I go in the recipe player, I have basically 2 options. If I click on open, then I can go in the recipe workroom, which is here. And then you can see all modules that were used for a recipe, and you can edit or cancel, for example, some modules. You can edit basically your recipe and you can save a new version. If I go back to the project, I have also the option export, and basically all modules will be displayed here in the project view. So not only the 2 that were selected, but now you can see all modules that were used for this recipe. Okay, so now I want to show you another workflow, which is always related to this project. So I will go to open another Amira project. So here, basically, these are the same. This is the same workflow. So before I showed you how you can use the HCS Plate Manager and how you can visualize volume rendering, which is here. So then, on the basis of this data, it was created a distance map. So basically, a distance map create an image which represents the distance inside the spheroid. And this is the representation. So the values in the center are more big compared to the values on the border. And on the basis of distance map, in parallel, it was analyzed the population of the T cells. So basically, it was calculated, so the object, the different structure were separated, some quantification analysis were done, and also the 3D surface of the population of the T cells, which is here. So now here you can see, so the population of the T cells. So on the basis of this analysis, distance map and the population of the T cells, it was calculated the penetration of the T cells into the spheroid, which is possible to observe here via color map. So you can see a different penetration distance on the basis of the color map of the different colors.